And I put my inoculation loop and some bleach, or alcohol, I mean. So what I'm gonna do next, I'm going to take my clamps, since I hadn't let the E. coli dry, air dry. Take my clamps, put the slide in there, and pass it through my fire 5 times. One, 2, 3, 4, and 5. So I'm gonna set that down and let it cool off. So I will do the same thing for my S. epidermis and S. cerevisiae, but for this video, I don't want it to be super duper long, like 30 minutes, because it's lengthy. So I'm just going to show you the E. coli, and off camera, I'll do the remaining 2. So once the slide has cooled, I will place it into my staining tray. Let me move my microscope out the way. I don't need it just yet. So I'll place my E. coli slide in my staining tray, and I will add 1 to 2 drops of the Crystal Violet Gram Stain 1. So 1, 2 drops is perfect. And there's that. So I will let that sit for 60 seconds. So it has been 60 seconds, so I'm going to take my E. coli and rinse it under tap water until it runs clear. And just let the excess go into the sink. Then I'm going to take my Gram Stain 2 and do the same thing. I will place 1 to 2 drops on there, and I will let that sit for 60 seconds as well. So PVP iodine. A little wet still. So 1, 2 drops of that, and I will let that sit for 60 seconds. Time consuming. In the staining tray, just hanging out. Okay, 60 seconds has gone by, so again, I'm going to rinse it. Excess water goes into the sink. There we go. So this will back on my staining tray. And then the Gram Stain 3, 2 drops. That will sit for 30 seconds. Well, one drop was enough, but it like spread, so my circle from my Sharpie looks gone. So maybe I should have put the circle on the other side. But at least I know where it's at. So 30 seconds. I think I'm gonna try to redraw the circle once I rinse it this time. Since you could still see the edges, it just kind of spread out into the same spot, it just spread out. But 30 seconds has gone by, so I'm gonna rinse that off. Drain the excess. So I have it at the bottom. I'm just gonna redraw my circle on the opposite side. So I'm gonna paste this back into the staining tray and do my gram staining for the San Fernin. 2 drops of that, and we'll sit on here for 30 seconds. 1 to 2 drops and 30 seconds. 1, 2. And it'll sit for 30 seconds. There you go. After that, I will rinse it again and place my side on my paper towel and let it dry completely. Then I'll place it on my microscope and view it at low, medium, and high magnification. And take a picture and stick that into my lab report. So 30 seconds has gone by, so I'm gonna rinse this. Take my slide, place on my paper towel so it can dry. To blotting the outside of the circle where I see water drops. I'm going to take my microscope back up here. Put this on the floor low. And place my slide. So under the Forex, it looks like everything's clumped together, but it looks like a puzzle where you see the outlines of the lines in the puzzle, like where you know what piece was what, but there's a line in there because the puzzle breaks apart. It looks like that, and it looks like a red purplish color. Ooh. On 10x, it's just way closer. And there's like, it looks like actually kind of like the earth. and like there's parts like rocks here and there in it that are bigger and stand more out. Kind of looks like it like 3D. And then 40. Just very up close. It looks like bacteria, but it looks like white and it outlines. Like you can't really tell, like it looks kind of like, it just looks dark. You can't tell if it's a red or purple. Even if that looks like that. It looks like something all come together and like there's pieces like a sponge, maybe. Like how there's holes and they don't all look the same. That's what it looks like. But yeah, I'm gonna go ahead and do the remainings for the S. epidermis and the S. cerevisiae, and I'll put that into my lab manual. And again, this is Siobhan Ferguson, Bio 225, week 3, and that is my lab experiment. Thank you.